Here is how the side counter will be set up for this demonstration. Before we begin, there is one thing to note for safety. Make sure you keep the ethanol away from the hot plate at all times. The hot plate will only be used to boil water. Pure ethanol boils at 78 degrees Celsius. The ethanol should only be heated to about 50 degrees Celsius in the water bath and not much higher. First, fill a beaker with distilled water and put it on the hot plate. Adjust the control knob to high in order to bring the water to a boil. Turn on the water bath and set the water temperature to 50 degrees Celsius. If the thermometer reads slightly higher than 50 degrees Celsius, that is fine, but make sure it doesn't approach 78 degrees Celsius, the boiling point of ethanol. Pour some pure ethanol into the glass beaker labeled ethanol and set this beaker in the water bath. Monitor the temperature of the water bath throughout the procedure. Check the temperature and then put the lid on the water bath. After you have prepared the water and ethanol, label the bench paper around the petri dishes where your leaves will be displayed. You will test 3 varieties of Coleus plants. The plants are, from left to right, green, maroon, and variegated. Pick 3 leaves from each plant. The maroon leaves here are easily distinguished because they have serrated edges. The green leaves and the variegated leaves, however, have rounded edges, so we will need to be able to distinguish between these two. So for our selection, we picked larger variegated leaves so that we would be able to distinguish them from the smaller green leaves after they had both lost their color. The variegated leaves are called variegated because they have a green edge to them. In the examples shown here, this green edge isn't as noticeable. However, if we part the superficial leaves of the variegated Coleus, we see leaves with a more pronounced green edge. This is an effect of receiving less sunlight. If necessary, you can pull the stems off of one of these 2 types so that you can distinguish between all of your leaves. Once your water is boiling, add the leaves to the water. Wait for the boiling water to leach out the anthocyanins. This should take about 10 minutes. Use a pair of forceps to push the leaves into the water. After the red pigments have been leached from the leaves, turn off the hot plate. At this point, you will still see some green and yellow coloring in the leaves from the chlorophyll. Place 1 anthocyanin leached leaf from each plant in a petri dish of water in its pre-labeled spot. Slowly open the lid on the water bath in order to keep the steam away from you. Place the other 6 leaves in the 50 degrees Celsius ethanol bath that you have prepared. Wait for the chlorophylls to leach out. This should take about 10 minutes. Again, do not allow the ethanol to boil. As the chlorophyll is leached from the leaves, the ethanol will turn green and the leaves will become white. Place 1 fully leached leaf from each plant in the pre-labeled petri dishes with some distilled water. Place the second fully leached leaf from each plant in a labeled petri dish and add distilled water to soften it. Saturate this second set of fully leached leaves with Lugol solution. The leaves should turn blue-black, indicating the presence of starch in them. Here's what the final demo should look like. Pour the beaker of water and anthocyanins down the sink. Remove the beaker of ethanol from the water bath and pour it into the solvent waste container. When finished with the leaves, throw them into the trash can. Pour the Lugols into the base waste container.